Now we are going to learn about special senses like taste and smell. Some learning objectives include try to list the types of taste and describe taste receptors, then trace the taste pathway and trace the olfactory pathway, then define disorders of taste and smell. So, some major terms are associated with this topic like gustation. Gestation is the sense of taste. Then olfaction, the sense of smell. Umami is a type of taste of food linked to glutamate content. Then the sense of taste and smell allow us to separate undesirable or even lethal foods from those that are pleasant to eat and nutritious. In addition, they elicit physiological responses that are involved in the digestion and utilization of foods. The sense of smell also allows animals to recognize the proximity of other animals or even individual animals. Finally, both senses are strongly tied to primitive emotional and behavioral functions of our nervous system. In this chapter, we discuss how taste and smell stimuli are detected and how they are encoded in neural signals transmitted to the brain. So, sense of taste. Taste is mainly a function of the taste buds in the mouth, but it is common experience that one's sense of smell also contributes strongly to taste perception. In addition, the texture of food, as detected by tactile senses of the mouth, and the presence of substance in the food that stimulate pain endings, such as pepper, greatly alter the taste experience. The importance of taste lies in the fact that it allows a person to select food in accord with desires and often in accord with the body tissue's metabolic need for specific substances. Then, primary sensations of taste. The identities of the specific chemicals that excite different taste receptors are not all known. Even so, psychophysiological and neurophysiological studies have identified at least 13 probable chemical receptors in the taste cells as follows like 2 sodium receptors, 2 potassium receptors, 1 chloride receptor, 1 adenosine receptor, 1 inosine receptor, 2 sweet receptors, 2 bitter receptors, 1 glutamate receptor, and 1 hydrogen ion receptor. So, for practical analysis of taste, the aforementioned receptor capabilities have also been grouped into 5 general categories called the primary sensations of taste. They are sour, salty, sweet, bitter, and umami. A person can perceive hundreds of different tastes. They are all thought to be combinations of the elementary taste sensations, just as all the colors we can see are combinations of these three primary colors. And sour taste. The sour taste is caused by acids, that is, by the hydrogen ion concentration. So, sour taste is caused by acids, that is, by the hydrogen ion concentration. And the intensity of this taste sensation is approximately proportional to the logarithm of the hydrogen ion concentration. The more acidic the food, the stronger the sour sensation becomes. Then, salty taste. The salty taste is elicited by ionized salts, mainly by the sodium ion concentration. The quality of the taste varies somewhat from one salt to another because some salts elicit other taste sensations in addition to saltiness. The cations of the salts, especially sodium cations, are mainly responsible for the salty taste, but the anions also contribute to a lesser extent. Sweet taste. The sweet taste is not caused by any single class of chemicals. Some of the types of chemicals that cause this taste include sugars, glycols, alcohols, aldehydes, ketones, amides, esters, and some amino acids and some small proteins, sulfonic acids, halogenated acids, and inorganic salts of lead and beryllium. Note specifically that most of the substances that cause a sweet taste are organic chemicals. It is especially interesting that slight changes in the chemical structure, such as addition of a sample radical, can often change the substance from sweet to bitter. Now, a bitter taste. The bitter taste, like the sweet taste, is not caused by any single type of chemical agent. Here again, the substance that give the bitter taste are almost entirely organic substance. Two particular classes of substances are especially likely to cause bitter taste sensations like Long chain organic substances that contain nitrogen and alkaloids. The alkaloids include many of the drugs used in medicine, such as quinine, caffeine, and strychnine and nicotine. Some substances that initially taste sweet have a bitter aftertaste. This characteristic is true for saccharin, which makes the substance objectionable to some people. Then the bitter taste, when it occurs in high intensity, usually causes the person or animal to reject the food. This reaction is undoubtedly an important function of the bitter taste sensation because many deadly toxins found in poisonous plants are alkaloids, and virtually all of these alkaloids cause an intensely bitter taste, usually followed by the rejection of food. And umami taste. Umami is a Japanese word meaning delicious. It designates a pleasant taste sensation that is qualitatively different from sour, salty, sweet, or bitter. Umami is the dominant taste of food containing L-glutamate, such as meat extracts and aging cheese, and some physiologists consider it to be a separate fifth category of primary taste stimuli. A taste receptor for L-glutamate may be related to one of the glutamate receptors that are also expressed in neuronal synapses of the brain. However, the precise molecular mechanisms responsible for umami taste are still unclear. Then threshold of taste. The threshold for stimulation of the sour taste by hydrochloric acid averages 0.0009 and or for stimulation of the salty taste by sodium chloride 0.01 m, for the sweet taste by sucrose 0.01 m, and for the bitter taste by quinine 0.00008 m. Note especially how much more sensitive the bitter taste sense is than the all others, which would be expected because this sensation provides an important protective function against many dangerous toxins in food. Relative taste indices, the reciprocals of the taste thresholds, are very important. The intensities of four of the primary sensations of taste are referred respectively to the intensities of the taste of hydrochloric acid, quinine, sucrose, and sodium chloride. Then taste blindness. Some people are taste blind for certain substances, especially for different types of thiourea compounds. A substance used frequently by psychologists demonstrate taste blindness is PTC or phenyl thiocarbamide, for which about 15 to 30% of all people exhibit taste blindness. The exact percentage depends on the method of testing and the concentration of the substance. Then taste bud and its function. Taste bud is of a diameter of about 1/30 mm and a length of about 1/16 mm. The taste bud is composed of about 50 modified epithelial cells, some of which are supporting cells called sustentacular cells and others of which are taste cells. The taste cells are continuously being replaced by mitotic division of surrounding epithelial cells. So some taste cells are young cells, others are mature cells that lie toward the center of the bud. They soon break up and dissolve. So when we try to learn about the parts of a taste bud, we could see nerve fibers will be there, taste cells will be there, subepithelial connective tissue is there, pores are there, microvilli will be there. Then stratified squamous epithelium region, along with microvilli, pore, nerve fibers, taste cells, subepithelial connective tissue lesions, etc. So the outer tips of taste cells are arranged around a minute taste pore. From the tip of each taste cell, several microvilli or taste hairs protrude outward into the taste pore to approach the cavity of the mouth. These microvilli provide the receptor surface for taste. Interwoven around the bodies of the taste cell is a branching terminal network of taste nerve fiber that are stimulated by the taste receptor cells. Some of the fibers that are stimulated by the taste receptor cells, some of these fibers invaginate into folds of the taste cell membrane. Many vesicles from beneath the cell membrane near the fiber. It is believed that these vesicles contain a neurotransmitter substance that is released through the cell membrane to excite the nerve fiber endings in response to taste stimulation. Then location of the taste bud. The taste buds are found on three types of papillae of the tongue. Like large number of taste buds are on the walls of struts that surround the circumvallate papilla, which form a villi in the surface of posterior tongue, and moderate numbers of taste buds are on the fungiform papillae over the flat anterior surface of the tongue, and moderate numbers are on the foliate papillae located in the folds along the lateral surfaces of tongue. Then additional taste buds are located on the palate, and a few are found on the tonsilla pillars, on the epiglottis, and even in the proximal esophagus. Adults have 3,000 to 10,000 taste buds, and children have a few more. Beyond the age of 45 years, many taste buds degenerate, causing taste sensitivity to decrease in old age. Then specificity of taste buds for a primary taste stimulus. Microelectrode studies from single taste buds show that each taste bud usually responds mostly to one of the 5 primary taste stimuli when the taste substance is in low concentration. However, at high concentration, most buds can be excited by 2 or more of the primary taste stimuli as well as by other taste stimuli that do not fit into the primary categories. So then other important points are also there, like mechanism of stimulation of taste buds, like receptor potential will be there. So the mechanism of taste cell, like that of most other sensory receptor cells, is negatively charged on the inside with respect to the outside. Application of a taste substance to the taste hairs cause partial loss of this negative potential, that is, the taste cell becomes depolarized. In most instances, the decreases potential within a wide range is approximately proportional to the logarithm of concentrations of the stimulating substance. This change in electrical potential in the taste cell is called the receptor potential for taste. The mechanism by which most stimulating substances react with the taste villi to initiate the receptor potential is by binding of the taste chemical to a protein receptor molecule that lies on the outer surface of the taste receptor cell near to or protruding through a villous membrane. This action in turn opens ion channels which allows positively charged sodium ions or hydrogen ions to enter the depolarize the normal negativity of the cell. Then the taste chemical is gradually washed away from the taste villus by saliva, which removes the stimulus. The type of receptor protein in each taste villus determines the type of taste that will be perceived. For sodium ions and hydrogen ions, which elicit salty and sour taste sensations respectively, the receptor proteins open specific ion channels in the apical membranes of the taste cells, thereby activating the receptors. However, for the sweet and bitter taste sensations, the portions of the receptor protein molecules that protrude through an apical membrane activate second messenger transmitter substance inside the taste cell, and this second messengers cause intracellular chemical changes that elicit the taste signals. Then generation of nerve impulses by the taste bud. On first application of the taste stimulus, the rate of discharge of nerve fibers from the taste buds arises to a peak in a small fraction of a second, but then adapts within the next few seconds back to a lower steady level as long as taste stimulus remains. Thus, a strong immediate signal is transmitted by the nerve taste nerve, and a weaker continuous signal is transmitted as long as the taste bud is exposed to the taste stimulus. Then the mechanism by which most stimulating substance react with the taste villi to initiate the receptor potential is by binding of the taste chemical to a protein in receptor molecule that lies on the outer surface of the taste receptor cell near to or protruding through villus membrane. This action in turn opens ion channels which allows positively charged sodium ions or hydrogen ions to enter and depolarize the normal negativity of the cell. Then the taste chemical is gradually washed away from the taste villus by the saliva, which removes the stimulus. Then the type of receptor protein in each taste villus determines the type of taste that will be perceived. For sodium ions and hydrogen ions, which elicit salty and sour taste sensations respectively, the receptor proteins open specific ion channels in the apical membrane of the taste cells, thereby activating the receptors. However, for the sweet and bitter taste sensations, the portions of the receptor protein muscles that protrude through the apical membrane activate second messenger transmitter substance inside the taste cell, and these second messengers cause intracellular chemical changes that elicit the taste signals. Then, generation of nerve impulses by the taste bud. On first application of the taste stimulus, the rate of discharge of the nerve fibers from the taste buds raises to a peak in a small fraction of a second, but then adopts within the next few seconds back to a lower, steady level as long as taste stimulus remains. Thus, a strong immediate signal is transmitted by the taste nerve, and a weaker continuous signal is transmitted as long as the taste bud is exposed to the taste stimulus. Then, transmission of the taste signals into the central nervous system shows the neuronal pathways for transmission of taste signals from the tongue and pharyngeal region into the central nervous system. Taste impulses from the anterior 2/3 of the tongue pass first into the lingual nerve, then through the cauda tympani into the facial nerve, and finally into the tractus solitarius in the brainstem. Taste sensations from the circumvallate papillae on the back of the tongue and from other posterior regions of the mouth and throat are transmitted through the glossopharyngeal nerve, also into the tractile solitarius, but at a slightly more posterior level. Finally, a few taste signals are transmitted into the tractus solitarius from the base of the tongue and other parts of the pharyngeal region by way of the vagus nerve. At all taste fiber synapse in the posterior brainstem in the nuclei of the tractus solitarius. This nuclei sends second order neurons to a small area of the ventral posterior medial nucleus of the thalamus, located slightly medial to the thalamic terminations of a facial region of dorsal column mediated lemniscal system. From the thalamus, third order neurons are transmitted to the lower tip of postcentral gyrus in the parietal cerebral cortex, where it curls deep into the sylvian fissure and into the adjacent opercular insular area. This area lies slightly lateral, ventral, and rostral to the area for tongue tactile signals in cerebral somatic area. From this description, the taste pathways, it is evident that they closely parallel the somatosensory pathway from tongue. Then taste reflexes are integrated in the brainstem. From tractus solitarius, many taste signals are transmitted within the brainstem itself directly into the superior and inferior salivary nuclei, and these areas transmit signals to the submandibular, sublingual, and parotid glands to help control the secretion of saliva during the ingestion and digestion of food. Then rapid adaptation of taste. Everyone is familiar with the fact that taste sensations adapt rapidly, often almost completely within a minute or so of continuous stimulation. Yet, from electrophysiological studies of taste nerve fibers, it is clear that adaptation of the taste buds themselves usually accounts for no more than about half of this rapid taste adaptation. Therefore, the final extreme degree of adaptation that occurs in the sensation of taste almost certainly occurs in the central nervous system. Mechanisms are not known. At any rate, it is a mechanism different from that of many other sensory systems, which adapts almost entirely at the receptors. So then, disorders of taste. Disorders of taste are either those that involve the loss of the ability to taste, or those linked to a distorted sensation of taste. Loss of the ability to taste is called ageusia or AGEUSIE. I'm not sure about the pronunciation. And while the reduced ability to taste is called hypogeusia. A distorted sense of taste is called dysgeusia. These conditions can occur due to advanced age or medications and constitute an important threat to the quality of life. Then, sense of smell. Smell is the least understood of our sense, partly because the sense of smell is a subjective phenomenon that cannot be studied with ease in lower animals. Another complicating problem is that the sense of smell is poorly developed in human beings compared with sense of smell in many lower animals. Then olfactory membrane. Before moving on to that, let's learn some important points about transmission of taste signals into the central nervous system, like gustatory cortex, anterior insula frontal operculum will be there. Then ventral posterior medial nucleus of thalamus will be there. Then tongue region, glossopharyngeal region, cauda tympani, geniculate ganglion. Then petrosal ganglion will be there. Then nucleus of solitary tract. Then gustatory area. Then nodose ganglion, and along with pharynx. Then organization of the olfactory membrane and olfactory bulb, and connection to the olfactory tract is important. Like olfactory tract will be there, olfactory bulb, mitral cell, Bowman gland, glomerulus, sustangula cells, olfactory cell, olfactory cilia, mucus layer. These are associated with that. So olfactory membrane, the histology of which is very important, lies in the superior part of each nostril. Medially, the olfactory membrane folds upward along the surface of the superior septum. Laterally, it folds over the superior subturbinate and even over a small portion of the upper surface of the middle turbinate. In each nostril, the olfactory membrane has a surface area of about 2.4 cm square. Olfactory cells are the receptor cells for smell sensation. The olfactory cells are actually bipolar nerve cells derived originally from the central nervous system. These are about 100 millions of these cells in the olfactory epithelium interspersed among sustangula cells, and the mucosal end of the olfactory cell forms a knob from which 4 to 25 olfactory hairs, also called olfactory cilia, measuring 0.3 μm in diameter and up to 200 μm in length, project into the mucus that coats the inner surface of the nasal cavity. Thus, projecting olfactory cilia form a dense mat in the mucus, and it is the cilia that react to odors in the air and stimulate the olfactory cells, as discussed later. Then, spaced among the olfactory cells and olfactory membrane are many small Bowman glands that secrete mucus onto the surface of olfactory membrane. Then, stimulation of the olfactory cells. Mechanism of excitation of the olfactory cells. The portion of each olfactory cell that responds to the olfactory chemical stimuli is the olfactory cilia. The odorant substance, upon coming in contact with olfactory membrane surface, first diffuses into the mucus that covers the cilia, and then it binds with receptor proteins in the membrane of each cilium. Each receptor protein is actually a long molecule that threads its way through the membrane about 7 times, folding inward and outward. The odorant binds with the portion of receptor protein that folds to the outside. The inside of the folding protein is coupled to a G protein, itself a combination of 3 subunits. Upon excitation of the receptor protein, an alpha subunit breaks away from the G protein and activates adenylyl cyclase, which is attached to the inside of the ciliary membrane near the receptor cell body. The activated cyclases in turn converts many molecules of intracellular adenosine triphosphate into cyclic adenosine monophosphate. Finally, the CAMP activate another nearby membrane protein, a gated sodium ion channel, which opens its gate and allows large numbers of sodium ions to pour through the membrane into the receptor cell cytoplasm. The sodium ions increase the electrical potential in the positive direction inside the cell membrane, thus exciting the olfactory neuron and transmitting action potentials into the central nervous system by way of the olfactory nerve. The importance of this mechanism for activating olfactory nerve is that it greatly multiplies the excitatory effect of even the weakest odorant. To summarize, activation of the receptor protein by the odorant substance activates the G protein complex, which in turn activates multiple molecules of adenylyl cyclase inside the olfactory cell membrane, which causes the formation of many times more molecules of CAMP, and finally, CAMP opens still many times more sodium ion channels. Therefore, even a minute concentration of a specific odorant initiates a cascading event that opens extremely large number of sodium channels. This process accounts for the exquisite sensitivity of the olfactory nerve. The importance of this mechanism for activating the olfactory nerves is that it greatly multiplies the excitatory effect of even the weakest odorant. To summarize, activation of receptor protein by the odorant substance activates the G protein complex, which in turn activate multiple molecules of adenylyl cyclase inside the olfactory cell membrane, which causes the formation of many times more molecules of CAMP, and finally the CAMP opens still many times more sodium ion channels. Therefore, even a minute concentration of a specific odorant initiates a cascading effect that opens extremely large numbers of sodium channels. This process accounts for the acquisite sensitivity of olfactory neurons to even the slightest amount of odorant. In addition to the basic chemical mechanism by which the olfactory cells are stimulated, several physical factors affect the degree of stimulation. First, only volatile substances that can be sniffed into the nostrils can be smelled. Second, the stimulating substance must be at least slightly water soluble so that it can pass through the mucus to reach the olfactory cilia. Third, it is helpful for the substance to be at least slightly lipid soluble, presumably because lipid constituents of the cilium are a weak barrier to non-lipid soluble odorants. Then membrane potentials and action potentials in olfactory signals and cells. The membrane potential inside unstimulated olfactory cells are measured by microelectrodes. Average is about negative 55 MV. With this potential, most of the cells generate continuous action potentials at a very slow rate, varying from once every 20 seconds up to 2 to 3% per second. Most odorants cause depolarization of the olfactory cell membrane, decreasing the negative potential in the cell from the normal level of negative 55 to negative 30 MV or less. That is changing the voltage in the positive direction. Along with this, the number of action potentials increase to 20 to 30 second inverse, which is a high rate for the minute olfactory nerve fibers. Then, over a wide range, the rate of olfactory nerve impulses change approximately in proportion to the logarithm of the stimulus strength, which demonstrates that the olfactory receptor obey principles of transduction similar to those of other sensory receptors. Then, rapid adaptation of olfactory sensations. The olfactory receptors adapt about 50% in the first second or so after stimulation. Thereafter, they adapt very little and very slowly. Yet we all know from our own experience that smell sensations adapt almost to extinction within a minute or so after entering a strongly odorous atmosphere, because this psychological adaptation is far greater than the degree of adaptation of the receptors. It is almost certain that most of the additional adaptation occurs within the central nerve system seems to be true for the adaptation of taste sensation as well. And the following neuronal mechanism for the adaptation is postulated. Large numbers of centrifugal nerve fibers pass from the olfactory regions of the brain backward along the olfactory tract and terminate on spatial inhibitory cells in the olfactory bulb and granule cells. It has been postulated that after the onset of an olfactory stimulus, the central nervous system quickly develops strong feedback inhibition to suppress relay of small smell signals through the olfactory bulb. Next topic we will be learning tomorrow.